This is the transporter and channel function for video. Big focus for this is front and center here, the channel proteins or channels, also known as ion channels if they're moving ions. And some of the things we're going to see in particular is about selectivity and channel gating. So here's the definition for channels or channel proteins. They are transmembrane proteins with a pore that allows small water-soluble molecules to cross the membrane. Some of the things that can vary across channel proteins are the pore size, which can be very wide. And for example, gap junctions contain very wide pores. But most of what we're going to talk about today is the narrow pore size. And so for narrow pore size, these are highly selective channels. Whereas for the wide pores, those are not very selective. An example of a channel with narrow pores we've heard about previously, and that's an aquaporin. And basically, the channels use passive transport. And as we've heard before, this means that the molecule that's being transported will be moving down its concentration gradient. Our focus is on ion channels, which is a type of channel protein which is going to move an ion through it. You can see one of the ion channel proteins here. This is a potassium channel. So the channel protein itself is in green, and here we see the membrane symbolized in gray. And then you can see that there's a potassium ion with a positive charge that's moving through this narrow region of the protein. There's two highly important features of ion channels. The first is that they are highly selective. And this is dependent on a few different things: charge, diameter of the pore, and finally the shape. And you can see that this is a special feature that's highlighted in this diagram, that there's a selectivity filter in this very, very narrow region here. And so there might be amino acids on either side of the selectivity filter that will be opposing charges to whichever ion is moving through. The second part about ion channels that is important for us to keep track of is that they are not continuously open. In other words, they're gated. And so there's going to be a few different stimuli that we'll hear about, each of which is dependent for the channel protein, that determine whether the particular ion channel is open or not. This figure on the right shows some more detail about what it means for the ion channel to be opened or closed. And I had an analogy to talk about for Gandalf. He's the wizard in The Lord of the Rings. And so a lot of times he's allowing movement across a bridge. And here he's going across the bridge with Bilbo, a hobbit, but anyway. And then there's another situation where Gandalf is going to prevent something from crossing a bridge, and so basically he's got this whole line about "you shall not pass," and it has nothing to do with grades. It is just kind of for fun to see how one thing, and in this analogy, Gandalf would be the protein, the ion channel. And it could have two very different properties for whether or not something can cross an area. So one thing that's confusing about this open and closed thing is, so the protein obviously has a different shape. When it's closed, it has this kind of gatekeeper going on. That's when the gate is closed, and then the ions cannot flow through. And when it's open, it has a different conformation, and then ions can move all the way through. An important thing to note about this change in conformation, which is again minor, and some books will say they do not change conformation, but there is obviously a shape change, and that's the confusing part. So basically, the ion channel does not change shape every single time that an ion moves across it. This is unlike, say, the sodium-potassium pump, which changes shape dramatically with each transfer of ions across the membrane. One important feature of this is that for ion channels, they are incredibly fast, so they can move ions amazingly quickly. You don't need to know these specific numbers, but just to impress you. So the ion channels can move 1 million ions per second. Each ion channel can move 1 million ions per second. On the other hand, the sodium-potassium pump can move a mere 500 sodium ions per second. So basically, ion channels are allowing for a much faster response to a stimulus, and they don't use ATP. And finally, let's talk a little bit more about what it's like for an individual ion to move across the membrane using a channel protein. So basically, here we're checking out the potassium ion. And it is normally in what's called a hydration shell when it's hanging out in a cell or an aqueous environment. So it is surrounded by many individual water molecules, which here are shown in blue. And so that's pretty large and pretty stable structure. As the hydration shell with the ion approaches the ion channel and gets down to where there's this narrow part, which is called the selectivity filter, the water molecules need to be stripped. And so basically the ion cannot fit through the selectivity filter with the hydration shell. And then after the ion passes through the selectivity filter, then it can start to recollect its water molecules and form a hydration shell again. And if you'd like to see a nice depiction of this, I would strongly encourage you to go check out movie 12.5 on the website for the textbook. And this shows a potassium channel, and it does a really good job of showing just how important the size is for specifically allowing potassium to go through the ion channel rather than other positively charged ions such as sodium. The next topic is to discuss how gated ion channels will open and close. And so there's 3 different types of gated ion channels, which are shown in this figure. And basically, let's start for each of these, and then we'll fill in below, so what determines whether each of these is open or closed. And so basically for this first one, again, we see these different colors for the ion channel proteins, and they're all in the membrane. And so outside of the cell is up, and the inside of the cell is down. And basically, for this voltage-gated channel, the determining factor is membrane potential. And you can see that in this first case, the ion channel is closed when the membrane potential is negative, and then it's open when the membrane potential is positive. And we will hear more about this later in a future topic, but an example of when this is used is in neurons as an action potential moves across the axon. Potential. Secondly, I'm going to put these 2 together. So there's ligand-gated channels. So ligand binding will determine whether each of these channel proteins is open or not. And basically, in one case, the ligand binds to the protein in the extracellular region, and in the other case, it binds to the protein in the intracellular region. An example of a type of ligand is a neurotransmitter, especially for these extracellular ligand-gated channels. Last but not least, we're going to talk about the mechanically gated ion channels. And so this type of channel protein will open depending on a physical force, which seems a little weird to imagine at first, but here's an example. So there are channel proteins which are present in the hair cells of the ear. And so this would be, again, membrane localized. And so those particular hair cells are going to receive a sound wave. And so they will physically move, and that triggers the opening of these channel proteins. And so this is by vibration of sound waves. And so you get the idea here that the ion channel proteins have a really key role in neuronal signaling and also in our sensory system, and as far as receiving information from external environment and then transmitting that forward.